Thank you everyone for spending your time at the seminar. We're just going to give it another minute for a few more attendees to come in, and then I will introduce our guest speaker. All right, so it is a great pleasure for me to introduce our final speaker from the stem cell and regenerative medicine theme as part of the leaders in the field. Today we have a great pleasure of hearing from Lydia Finley. So Lydia went to Yale for her undergraduate, where she studied biology and did metabolism research as an undergrad with Dr. Neufer. She spent a year teaching science at the Taft School in Connecticut before moving to Boston to get her Ph.D. at Harvard in the biological and biomedical sciences program. And there she studied with Dr. Marcia Haggis and did more work on metabolism, studying sirtuins and caloric restriction. After her PhD, she moved to New York, her home city, where she did her postdoctoral fellowship with Dr. Craig Thompson, where she made some really enormous discoveries about metabolism in stem cells and relationship to epigenetics. She then was hired to become an assistant professor at the Gerstner Sloan Kettering Graduate School, as well as at the Weill Cornell Graduate School, and she's an assistant member in the Sloan Kettering cell biology program. Her research has continued in this area of stem cell biology, metabolism, and epigenetics, really making important discoveries connecting nutrient metabolism with gene expression, cell fate, and differentiation. It's inspired a lot of people in the field. It's certainly inspired my own research in our lab. Dr. Finley has been the recipient of a lot of awards and honors and fellowships. So in graduate school, she was a NSF graduate research fellow. During her postdoc, she had a Damon Runyon fellowship. In 2018, she became a Searle Scholar, and in 2020, she won an award for the Pershing Square Stone Prize for Young Investigators in Cancer Research. So she has accomplished a great deal and has a whole lot more ahead, and we're very excited to hear today's seminar from Lydia Finley. Thank you.
Thank you so much, Andrew, for the really generous introduction and the invitation to be here today. Thank you to you and April. I have to say, I can't think of an institution that has more people in it that study things that I'm interested in than UCLA. I think the question that my lab is really interested in, how metabolites control cell fate and how metabolic pathways change as cells undergo developmental changes, is something that a lot of people at UCLA are doing really amazing work on. And so it's been a lot of fun to speak with some people today, and I hope to continue speaking to people moving forward. So broadly speaking, we are interested in the reciprocal relationship between metabolism and cell fate decisions. And so in particular, we think about this in a developmental context where stem cells become progenitor cells and ultimately start to specify and become differentiated cells. I think it's increasingly appreciated that these fate transitions are accompanied by metabolic transitions, and that cells in different stages of development or identity have different metabolic profiles. We think this is a really interesting question to identify the metabolic pathways that characterize specific stages of development and to understand what drives these metabolic changes for a couple of reasons. The first being, if we can begin to try to understand the metabolic pathways that characterize cells in different stages of development, perhaps we can devise strategies to select for or enrich for cells with desired phenotypic properties. Another reason that we think understanding the metabolism that accompanies sulfate transitions is important is because of the hypothesis that maybe these metabolic changes do more than just accompany differentiation, but might actually drive them. That metabolic changes might shape the propensity of the cells to undergo a sulfate transition and change its identity. So how might metabolites affect sulfate transitions? There are, I think, several hypotheses. Metabolites are known allosteric regulators of many pathways. They, of course, directly activate signaling pathways such as TOR and AMPK. But one pathway, one hypothesis that we are particularly interested in the lab is the idea that metabolites might control the ability of cells to regulate their gene expression programs. Because ultimately, in the end, gene expression programs are what define the identity of cells. And to change your lineage, you have to change your gene expression program. And in many cases, the expression of particular genes is regulated in part by the modifications of the histones and the DNA itself that are in that neighborhood of the gene. So these chromatin modifications that surround genes can include repressive modifications such as methylation on histones or DNA, or activating modifications such as acetylation or other acylations on histone lysine tails. And the relationship between metabolism here is actually, I think, a little bit intuitive. These modifications are themselves metabolites. So for example, methyltransferases use the metabolite S-adenosylmethionine as the obligate donor for methylation reactions for the deposition on chromatin. Analogously, histone acetyltransferases use acetyl-CoA as the obligate acetyl donor for histone acetylation. Often the removal of these modifications also depends on metabolites, and this is best exemplified, I think, by a family of proteins called alpha-ketoglutarate-dependent dioxygenases. It's a large family of enzymes that includes many chromatin-modifying enzymes, including histone lysine demethylases and the TET family of methylcytosine oxidases that can iteratively oxidize 5-methylcytosine to hydroxymethylcytosine, ultimately leading to the removal of this repressive modification. These enzymes consume alpha-ketoglutarate and oxygen as obligate co-substrates and produce carbon dioxide and succinate as products. So I hope what you can appreciate here is that metabolites are really integral to many of the reactions that control the chromatin landscape and gene expression profile. And these aren't just sort of random metabolites. These metabolites sit at really central nodes of intracellular metabolism. In particular, many of the metabolites that control chromatin modifications are intermediates of the tricarboxylic acid, or TCA cycle, a suite of biochemical reactions that sits in the mitochondria and is responsible for consuming our major nutrients like glucose and glutamine. It oxidizes these nutrients and producing the energy, the reducing equivalents, and the oxidized substrates that are needed to fuel growth. So we like to think of the TCA cycle as a real integrator of cell state. It's consuming nutrients, breaking them down, and it's producing the products that are required for cells to regulate either homeostatic or proliferative metabolism. And so at this central integrating metabolic node, many of these metabolites that are intermediates of this pathway also can, if they can leave the mitochondria, contribute to regulation of the chromatin landscape. This raises, I think, a really exciting possibility that metabolic control of gene expression might be a very elegant way for cells to couple the integral metabolic pathways, their nutrient availability, their nutrient demand, with the gene expression profiles that determine cell fate. So to address the relationship between metabolism and cell fate, a model system that we have found to be extremely useful in this regard are mouse embryonic stem cells. Mouse embryonic stem cells, I think, are a really great model for cell fate transitions. I know many people at UCLA are doing amazing things with them. And traditionally, or not traditionally, mouse embryonic stem cells are, by definition, derived from the pre-implantation epiblast of the pre-implantation blastocyst. These cells can be captured. The pluripotent state, I should say, traditionally is transient in a normally developing blastocyst, but it can be captured in vitro by culturing cells traditionally in a medium formulation that contains serum and leukemia inhibitory factor, or LIF. Get that out of the way. When embryonic stem cells are cultured in this medium, they are pluripotent. They can give rise to all adult lineages. They can even give rise to an entire mouse. But they are functionally heterogeneous, which is best seen by immunofluorescence staining for a core pluripotency transcription factor NANOG, shown here in red. You can see that some cells have high NANOG and some cells have low NANOG. This corresponds to functional heterogeneity, such that the cells with higher nanog are more prone to self-renewal, whereas the cells with lower nanog are more prone to differentiation. So while this medium formulation allows you to capture an otherwise transiently existing pluripotent state, it is complicated by the heterogeneity and metastability of this culture condition. Around 13 years or 14 years ago, so now, Austin Smith's lab in Cambridge in the UK came up with an alternative medium formulation that really solves the issue of this metastability. In this traditional formulation, serum is replaced by two inhibitors, two kinase inhibitors, an inhibitor against GSK3 beta and an inhibitor against MEK-ERK. When cultured in the presence of these two inhibitors, or 2I, the cells are now much more homogeneous, which you can again see by staining for nanog, which is now much higher and much more uniform. These cells are thus much more resistant to spontaneous differentiation. They're much more likely to self-renew. And importantly, if you compare the gene expression and epigenetic profiles of these cells, they look very similar to the pre-implantation epiblast cells, arguing that these cells represent a sort of naive ground state of pluripotency that represents the earliest state of pluripotency that can be reproducibly captured in a dish. We love these two media formulations because they provide us an opportunity to study the relationship between metabolism and cell fate while stripping out many of the confounding variables that can often clutter our attempts to uncover the links between metabolism and fate. And that's because we can take the exact same cell type, the exact same mouse embryonic stem cell line, and we can grow it in either medium formulation, and the cells will proliferate at the same rate, and we can control or begin to control many of the variables in the media, isolating them down to as few variables as possible, therefore allowing us to begin to understand the metabolic changes that accompany this particular change in cell fate, that accompany the propensity for naive self-renewal from changes in metabolism that might be due to changes in media formulation or changes in growth rate or changes in cell size, for example. And so armed with this well-controlled system to study metabolism in cells with slightly different cell fate potential, we a few years ago began just by looking at the TCA cycle, again, because this is a metabolic hub that we think of as being a really important integrator of cellular metabolic demands. This work was all done in collaboration with Dave Alice's lab back when I was a postdoc, and in particular the postdoc Bryce Carey, who actually taught me how to culture ESCs way back in the day. So starting really basic, just by comparing cells cultured in serum and leukemia inhibitory factor, or 2I, we looked at how cells in these different conditions use nutrients. We started by looking at the most abundant nutrients in the medium, glucose or glutamine, by giving cells heavy labeled isotopic versions of each of these nutrients, and then asking how these nutrients are incorporated into the TCA cycle in proliferating ESCs. When we looked at glucose, we found that cells cultured in the naive ground state, shown in blue, put many more glucose-derived carbons into the TCA cycle, showing here two representative metabolites, malate and glutamate. They put these glucose-derived carbons into the TCA cycle faster and to an overall higher level than their serum lift cultured counterparts. This was accompanied by a reciprocal decrease in the utilization of glutamine. So when we looked at the ability of cells to use glutamine to fuel the TCA cycle, we found that the naive cells put fewer glutamine-derived carbons into the TCA cycle. Shown here again is a slower and lower labeling of glutamine-derived carbons to malate and glutamate. So this argued that there might be a substrate switch occurring when cells entered the naive ground state, such that they were less reliant upon glutamine, which is traditionally the most important carbon source for TCA cycle metabolites in cells in culture. And indeed, this metabolic shift actually seems to be functional. If we removed glutamine from the medium, when we cultured serum lift cells in the presence of serum and lift, they were incapable of growing. And this is pretty much what you would expect for almost any mammalian cell in culture. Most mammalian cells in culture cannot grow in the absence of glutamine, and they often begin to die in the absence of glutamine. Again, because it's this critical source of carbon for the TCA cycle, for nitrogen for nucleic acid biosynthesis, etc. I should also mention that every line you're looking at here is a different embryonic stem cell line. If we took these 4 independent embryonic stem cell lines, though, and now we cultured them under naive conditions, we found that they could grow in the absence of exogenous glutamine. There's no glutamate here either, hardly any non-essential amino acids, and yet these naive embryonic stem cells could undergo successive population doublings despite the absence of exogenous glutamine. This was exciting to us, and it argued that the findings that we'd had just from our isotope tracing data really did predict functional difference, functional dependencies of these embryonic stem cells on glutamine metabolism. So to test whether or not this difference in glutamine metabolism was intrinsically because of the identity of the cell or because of some of these changes in culture conditions, we took advantage of the inherent heterogeneity of cells cultured in serum lymph. To do this, we made use of a reporter generated by Rudolf Janish's lab, in which a GFP reporter has been knocked into the endogenous nanog allele. This allows us then to read out just by flow cytometry a heterogeneous population shown in serum lymph, shown in gray. You see that there's a wide distribution of nanog expression, and you see they form a bimodal population with nanog low and nanog high cells. If we culture these cells in the presence of 2I, now they become highly homogeneous with high levels of nanog. So with this heterogeneous population, we could then sort out the lowest nanog expressing cells or the highest nanog expressing cells and assess their metabolic demands functionally. What we found is that the low nanog expressing cells were extremely prone to apoptosis upon glutamine withdrawal, but the nanog high cells were much more resistant to apoptosis upon glutamine withdrawal, approaching the levels of resistance that cells had in the presence of 2I. So this suggested to us that as cells began to commit, or as cells began to exit the highly self-renewing state, they became more dependent upon exogenous glutamine. So to test this another way, we turned to staining for OCT4, which is a much more critical marker of pluripotency than nanog. Low nanog means you're prone to differentiation, but it doesn't mean you're committed to differentiation. In contrast, if you've lost OCT4, you have exited the naive pluripotent state. And what we see here when we culture cells in serum and lymph is that there's a wide distribution of cells such that a high proportion of them actually have quite low OCT4 expression. These are the cells that are exiting the pluripotent state. If we withdraw glutamine, we see a time-dependent decrease in this number of OCT4 low cells, indicating that losing glutamine results in selective death of the cells that have exited the naive pluripotent state, such that at the end of 24 hours after glutamine withdrawal, the cells that are left are much more likely to be cells that are in the naive pluripotent state. And so we reasoned that then if we could restore glutamine, add glutamine back after this pulse, this 24-hour deprivation of glutamine, we could therefore have a population that now is much more enriched for naive pluripotent cells. This is exactly what we see. Now I'm showing you with the nanog GFP reporter, that in control conditions, there's a shift, there's a balance of nanog low and nanog high cells. But if we've subjected cells to 24 hours of glutamine deprivation and then restored glutamine, now the population is much more likely to be nanog high. We've enriched for a population of highly self-renewing stem cells. So where might this ability to shift the population, the self-renewal potential of a population, have utility? We reasoned that reprogramming might be an area where this could have a lot of utility, theoretically, because reprogramming, especially of mouse cells, is inherently extremely slow and inefficient. And one place where there's a lot of inefficiency is sort of these terminal stages of reprogramming, where many cells have begun to look like they've entered the pluripotent state, but they haven't truly reactivated the endogenous pluripotency transcriptional network. So to assess for reactivation of the endogenous pluripotency network, we took advantage of cells which have an OCT4 GFP reporter now. Turning on of this marker means the cells have reactivated that endogenous pluripotency network. And we had them undergo traditional OKSM reprogramming, but just at the very end, we then subjected cells to 24 hours of either glutamine withdrawal or 2I treatment, or we gave them 2I throughout the course of the experiment. What we found is that because glutamine withdrawal selectively depletes cells that aren't truly endogenously pluripotent, we were able to purify the population, and therefore enrich for cells that had truly completed the reprogramming process. And I'd like to note that the 24 hours of glutamine withdrawal, that was it, no drugs, no nothing, was enough to look almost as good as adding 2I, these sort of gold standard inhibitors that help cells enter the naive pluripotent state, which I think argues that metabolism isn't just a sideshow to signal transduction, but if we really can understand the metabolic networks that are required for the propagation and identity of specific cell types, we can design interventions that are at least co-equal with signal transduction for inducing cells with desired phenotypic properties. So naive stem cells can grow without glutamine. And we think that's useful. We think you can use that different nutrient dependency to shift the balance of your population. But there's also a fundamental biological question here, which is how do naive embryonic stem cells grow without glutamine? They have to make it, clearly. Glutamine, as I mentioned, is essential. It's the essential nitrogen donor for nucleic acid biosynthesis. It does a lot of important things for amino acid exchange, as shown by Heather's group. But also, of course, you need all your amino acids in order to make protein. So if cells are growing without glutamine, that means they are making it. And glutamine can be made from a carbon backbone of an intermediate, TCA cycle intermediate alpha-ketoglutarate. Alpha-ketoglutarate can be aminated and amidated to form glutamine. And in cancer cells, I would say some of these latter steps seem to be limiting for cells and be the drivers of their real glutamine dependence. What we found in embryonic stem cells is that it's the carbon backbone itself that's limiting for glutamine formation. And the way we could show this is that if we withdrew glutamine from our metastable embryonic stem cells, the cell cycle dramatically slowed, which is shown here as an increase in doubling time, and the cells would eventually die. But we could completely rescue this by adding any form of cell-permeable alpha-ketoglutarate, a dimethyl, a diethyl, whatever. If you add alpha-ketoglutarate, that is sufficient now to maintain normal proliferation and viability in serum lymph cells, arguing that it's the carbon backbone that is limiting for glutamine independence. And so we wondered if the ability of cells growing in the naive state to survive without glutamine was because they could actually accumulate alpha-ketoglutarate in excess of its need to be oxidized by the TCA cycle. So to test this, we looked at 4 different embryonic stem cell lines cultured in serum lymph for 2I. And we found that whenever we looked at levels of alpha-ketoglutarate relative to downstream metabolites in the TCA cycle, shown here as succinate as an example, but this will hold with fumarate, malate, aspartate, whatever, there was always more alpha-ketoglutarate than these oxidized downstream metabolites, suggesting that the naive cells are capable of allowing alpha-ketoglutarate to accumulate in excess of its need to be oxidized by the TCA cycle. And again, this isn't just a trick of 2I, it appears to be an intrinsic feature of cells in the naive ground state of pluripotency, because if we sort out the nanog low and nanog high cells, we again see this disconnect between alpha-ketoglutarate levels and levels of its downstream oxidized metabolites, such as succinate. So altogether, this told us that there are intrinsic metabolic differences between cells depending on their self-renewal potential, one of which is an increase in intracellular alpha-ketoglutarate. So what is the consequence of that? If you are accumulating alpha-ketoglutarate in excess of your need to oxidize it, what does that mean for cells? Well, this gets back to the initial idea that I started out with, that some of these metabolites that are TCA cycle sort of sentinels might contribute to regulation of the chromatin landscape. And indeed, there are those family of alpha-ketoglutarate dependent dioxygenases that consume alpha-ketoglutarate and are competitively inhibited by succinate that regulate the chromatin landscape. So that led us to the hypothesis that if cells could accumulate alpha-ketoglutarate at the expense of succinate, you might actually potentiate the ability of these enzymes to act. You might potentiate the ability of the TETs to iteratively oxidize methylcytosine, or the Jumanji domain containing histone demethylases to remove lysine modifications. We thought that this was a relevant hypothesis in embryonic stem cells because differences in chromatin methylation are well known to accompany cell fate transitions. For example, mouse embryonic stem cells grown in serum and lymph actually have high levels of DNA methylation, reminiscent almost of somatic counterparts, whereas cells cultured in the naive ground state are known to have global DNA hypomethylation. Given that these cells also had alpha-ketoglutarate, we wondered if this metabolic state might be one factor contributing to the hypomethylated landscape of naive embryonic stem cells. To test this, we simply added cell-permeable alpha-ketoglutarate to the methylated serum lymph-cultured ESCs, and we looked at levels of DNA methylation. We've done this a number of ways, most recently by LCMS with Tron He's group at University of Chicago. And what we see is that cell-permeable alpha-ketoglutarate alone is sufficient to induce a really robust loss of DNA methylation, almost 50%,
over the course of just a couple days of culture. So this argues to us that the metabolic differences observed in these cells might contribute to regulation of the chromatin landscape. And given the tight correlation between chromatin modifications and self-renewal, we next asked if this had any implications for the ability of the cell to engage in naive self-renewal. To do this, we again just supplemented the metastable embryonic stem cells with cell-permeable alpha-ketoglutarate, and we found that this is again sufficient to shift the population of the cells. Just by adding alpha-ketoglutarate now, we can see many more cells are in the nanog GFP high population. This is accompanied by an increase in self-renewal potential. If we see these cells at single cell density in the presence of alpha-ketoglutarate, they are much more likely to form tight, red, alkaline phosphatase positive colonies indicative of self-renewal, whereas cell-permeable succinate does not do this. So overall, this tells us that in cells reminiscent of the pre-implantation epiblast, maintenance of high levels of alpha-ketoglutarate relative to its downstream metabolites can potentiate self-renewal. And so we've been really interested in the question of how this happens. And it's not as trivial as maybe we thought it was, and we've spent many years on it, but I'll tell you some of our thoughts on it, and hopefully in a few years we'll have more updates for you. But to best we can reason, there are potentially 3 ways that alpha-ketoglutarate could orchestrate fate changes. First, maybe it could amplify the activity of existing transcriptional networks. So say it facilitates demethylation, it might help, say, nanog bind the regions that nanog needs to activate, for example, and therefore amplify the ability of nanog to do its job. It's one possibility. Another possibility is that it might shield cells from responding to inductive cues. Perhaps by sequestering transcriptional coactivator machinery at active sites, you would prevent them from being redistributed maybe to where they would need to go to turn on a new gene expression program. And the third hypothesis that we could come up with is that by forcing demethylation, alpha-ketoglutarate would prevent the silencing of genes that you need for a fate change. So for example, as you exit the naive pluripotent state, you have to downregulate pluripotency transcription factors such as nanog. And maybe the alpha-ketoglutarate antagonizes that silencing. I should say that this hypothesis was given a big boost by really beautiful work from Michael Title's lab here at UCLA, published just shortly after our work on alpha-ketoglutarate, done by Tara Tesla, showing that looking in a slightly more committed state of development in human embryonic stem cells, alpha-ketoglutarate actually accelerated lineage commitment. And we were really excited about this beautiful work because it seemed to suggest, and indeed they did suggest, point 3, that maybe alpha-ketoglutarate helps, by facilitating demethylation, helps prevent these fate changes. And just to show you a little bit of data from our systems where we think that that might be true, we can take advantage of this nanog GFP reporter and look at how cells respond to stresses that would induce differentiation or exit of the naive pluripotent state. We can add retinoic acid, we can remove LIF, et cetera, et cetera. All of these stresses will collapse the nanog high population in control cells. But whenever we treat the cells with alpha-ketoglutarate, we now can really maintain for longer than I would think possible a population of nanog high cells, arguing that alpha-ketoglutarate really does, in this context, help lock you into this naive pluripotent state. So we're also interested in this idea that if alpha-ketoglutarate can potentiate demethylation, how this might affect fate changes in other systems. And one system that we've become quite interested in are adult stem cells. The reason we're interested in this is that in many ways it's an inverse chromatin problem as naive pre-implantation epiblast cells. Whereas the pre-implantation epiblast cells have to methylate their pluripotency factor genetic regions to begin to exit the pluripotent state. Adult stem cells actually have to demethylate lineage specifying regions. So these genes that ultimately allow them to become a committed differentiated cell have to be demethylated and activated. And so we had the hypothesis that maybe alpha-ketoglutarate could potentiate differentiation in some adult stem cell lineages. This question was asked by Sanji Bash, a really talented MD Ph.D. student jointly mentored by myself and Elaine Fuchs at the Rockefeller University, and Pavlina Todorova, a postdoc in my lab. Their reasoning along with Elaine Fuchs, our amazing collaborator, was that epidermal stem cells are a really good tissue to ask this question. Epidermal stem cells sit here at the epidermal-dermal junction. They can differentiate and give rise to the committed cells that form our skin barrier. And importantly, with stresses, these cells can acquire mutations that prevent them from activating terminal differentiation and therefore lead them to be likely to form tumors such as squamous cell carcinoma. And it seemed like a reasonable hypothesis that metabolism might affect the self-renewal potential of these epidermal stem cells because of work from Elaine and others' labs who have shown that loss of one methylation mark in particular, histone H3 lysine 27 trimethylation, is really important in epidermal differentiation. So what has been shown is that activation of alpha-ketoglutarate-dependent dioxygenases can facilitate removal of this chromatin methylation and in a manner that potentiates differentiation. And conversely, enforced methylation, say through expression of the methyltransferase EZH2, which is fueled by the metabolite acetylmethionine, can maintain high levels of this mark and promote self-renewal. And so the question we wanted to know is, do the metabolic profiles of epidermal stem cells affect their ability to regulate this chromatin modification that controls their cell identity? So we did what we always do, which is we take stem cells, in this case epidermal stem cells, and we gave them labeled forms of the most abundant nutrients, glucose and glutamine, and we looked at how the cells fueled their TCA cycle from these abundant nutrients to see how the TCA cycle might be communicating to the nucleus to regulate gene expression. It was a good idea. What we found was that the TCA cycle was changing, sure, but the biggest change we saw, the change we couldn't ignore, much to my chagrin, was a huge induction in the ability of these cells to make serine. Epidermal stem cells just made a ton of serine from glucose, and we've done glucose tracing in a lot of cell lines. So it jumped out to us quite clearly that this was a lineage that for some reason, and maybe it's the requirement to make sphingolipids as part of the epidermal junction, and serine is an important component of that, whatever reason, epidermal stem cells make a lot of serine. And that was exciting to us, because serine has been shown by Jason Locazal and Karen Bowson and others to be a source for this metabolite acetylmethionine that can deposit, that is the required donor for methylation. So we were excited that maybe we had come to a different route for metabolic pathways to regulate sulfate in the epidermis, and we immediately went to test whether or not starving cells of serine and its closely related metabolite glycine could affect this critical chromatin modification H3K27 trimethyl. That's exactly what we saw. If we took epidermal stem cells and we removed serine and glycine, we saw a loss of H3K27 trimethyl, shown here in purple. Now, we then reasoned that if cells were making so much serine, we could inhibit flux through the serine synthesis pathway. I think my animations are out of order, sorry. We could inhibit flux through the serine synthesis pathway, and that would starve cells of serine perhaps even in the control conditions, and maybe therefore knocking down PHGDH would reduce H3K27 trimethyl even in the presence of serine. You can see that's not what we saw. In fact, what we saw was that blocking flux through the serine synthesis pathway actually prevented loss of H3K27 trimethyl, even though now in this condition, with no serine exogenously, oops, my animations are out of order, sorry. With no serine exogenously and no way to make serine, intracellular serine was even lower. So this told us that this methylation mark was actually not regulated by the presence or absence of serine at all, but rather was regulated by activation of flux of the serine synthesis pathway. If you starve the cells of serine and glycine, they activate this pathway even more, and that causes demethylation, which is blocked if you can't activate flux of this pathway. And the critical byproduct of this pathway that's responsible for this is alpha-ketoglutarate, which again, I'll skip over a lot of experiments. I did not like this hypothesis, but if anyone wants to hear how Sanji convinced me that it's true, I'm happy to tell that story. In any case, we can completely rescue the effects of PHGDH silencing by adding this product of the serine synthesis pathway, alpha-ketoglutarate. So this is arguing to us that de novo synthesis of serine induced by serine starvation could allow cells to accumulate alpha-ketoglutarate, which potentiated loss of this histone methylation and also induced differentiation. So we found that when we starved epidermal stem cells of serine, they differentiated in the dish, which you can see here by loss of the stem cell marker, keratin 14, shown in green. If we blocked flux of the serine synthesis pathway, They no longer differentiated, even if they were starved of serine. And importantly, we could completely restore differentiation in the presence or absence of serine just by adding alpha-ketoglutarate. We also could see this in vivo. If we put mice on a serine glycine-free diet, we actually saw that their epidermis would begin to induce signs of precocious differentiation and would actually thicken, which is pretty cool. So that told us all together that the source of serine was really important for the regulation of cell fate of these epidermal stem cells, such that if serine was available to them to take up, they were more likely to self-renew. But if they were starved of serine, they were more likely to undergo terminal differentiation. And so given the fact that differentiation is a major tumor suppressive mechanism in the skin, we asked if the source of serine available to cells after they'd acquired an oncogenic mutation might shape the likelihood of whether or not a cell forms a tumor. Because I think a really interesting finding that's come out of these big whole cell, single cell sequencing experiments in human tissue is that we are all riddled with oncogenic mutations, especially as we age. And yet, thankfully, we don't always become balls of tumors. So there's something else besides oncogenic mutations that control the ability or the likelihood of a cell to form a tumor. We're interested in the idea that metabolism might be one component in that might shape the propensity for differentiation. So to test this idea, we put mice on a serine glycine-free diet for 2 weeks, and then we performed a tumor initiation experiment by treating them with these nasty combination of chemicals that will induce mutations in the epidermal stem cells that will allow them to form squamous cell carcinomas. We found that starving mice of serine and glycine delayed tumor initiation, delayed tumor onset, and decreased the total number of tumors that were formed, indicating that serine glycine starvation is indeed a manner to slow and reduce tumor initiation. We could also do this in established tumors by forming allografts. So we could take squamous cell carcinoma lines, inject them subcutaneously into mice on a control or serine glycine-free diet. And we found, as other people have shown, serine glycine starvation reduces growth of xenografts and allografts. That's not new. We saw really dramatic change, though, in tumor growth. This is a huge, huge, huge change in tumor growth. And we think, again, that has something to do with this lineage. These epidermal cells really, really, really like to make serine. And so they're very sensitive to it. We found that the tumors that do form, they're quite small, but the tumors that do form look different. They have less H3K27 trimethyl, and less, shown in purple, and less of that stem cell marker, keratin 14, shown in white. Here's another picture on this. Cancer cells are all labeled red, and the keratin 14 is shown in white. We see loss of this stem cell marker. We also see changes in the number of stem cells in the tumors themselves. Shown here in CD44 are the tumor-associated stem cells, which sit here along the edge of the tumor, and there are many fewer of them once the tumors are starved of serine glycine. So why does serine slow tumor growth? Well, again, it's not because of the presence or absence of serine itself. It's because of activation of flux of the serine synthesis pathway. So if we inject cells that now have silencing of Ph.D, they cannot activate serine synthesis even when they're starved of serine and glycine. And thus these tumors, although they're small, they're very small because they don't have any serine to grow, they can't differentiate. So they're locked in this state. And what that means actually is that if you look at the stem cell compartment of the tumor, they're just chock full of stem cells. These cells can't differentiate, and they're just sitting around these little tumor-associated stem cells, which actually led us to wonder if maybe serine synthesis or loss of activation of serine synthesis could be tumor suppressive. So if we just quantify the number of tumor-associated stem cells, we see that even in control cells, there's a big decrease in these number of tumor-associated stem cells, indicating that there is some sort of basal serine synthesis that can be activated in the cells. And if we knock out Ph.D, we have many more stem cells, even on a controlled diet, arguing again that even on a controlled diet without any intervention, there is some serine synthesis going on that can be tumor suppressive. And what that means then is if even on a controlled diet, some serine synthesis is going on that can be tumor suppressive, if you look at the very earliest stages of tumor initiation, the very earliest stages of tumor growth, silencing Ph.D actually can be oncogenic. You can have a growth advantage early until the tumor becomes so big that it really, really needs to go find some serine, and it's not happy that it can't make it. So how does serine starvation induce differentiation? Well, we were working again under the hypothesis that it was because of induction of alpha-ketoglutarate, a byproduct of the serine synthesis pathway. So to be able to manipulate alpha-ketoglutarate and the competitive antagonist succinate genetically, we engineered hairpins against oxyglutarate dehydrogenase, which consumes alpha-ketoglutarate, or succinate dehydrogenase, which consumes succinate. So these are ways to enforce high levels of alpha-ketoglutarate or succinate, respectively. If we take these genetically modified lines, inject them subcutaneously into mice, allow them to form tumors, and look at what happens to the tumor, we see that, again, control cells on a control diet are full of undifferentiated cells shown in white. But when these mice are put on a serine glycine starvation diet, they differentiate, which you can show here by gain of the terminal differentiation marker keratin 10 shown in red. If we force high levels of alpha-ketoglutarate, these tumors actually show differentiation even on the control diet. And if we force high levels of succinate, these tumors are resistant to differentiation induced by serine glycine starvation. So they do not differentiate. And what that means is that while messing with your TCA cycle isn't great for tumor growth on a control diet, you can see knocking down any TCA cycle enzyme has a fitness disadvantage in normal conditions. There's a big difference when you put the mice on a serine glycine-free diet. This complete obliteration of tumor growth that we see when we grow tumors on a serine glycine-free diet is completely blocked in the presence of high levels of succinate. So these tumors do not differentiate, and therefore they do not experience the tumor suppressive effects of serine glycine starvation. So that's just an example of sort of the questions that we're interested in, of how, by starting by first principles of understanding what is the metabolic phenotype of a cell, how does it use its nutrients, and what does that mean for intracellular metabolite levels, we can start to understand how the metabolic profiles of cells can influence their propensity for fate change. And in the case of embryonic stem cells or in epidermal stem cells, alpha-ketoglutarate seems to be one of, I'm sure, many, many, many players that can shape the likelihood of a cell to differentiate or self-renew. But just to show you a little bit of where we're going this in the last few minutes, some very preliminary work, we've become struck by the fact that both of these stories came from the fact that we always trace glucose and glutamine when we start to look at different cell systems. And so in a way, we're biased as to what we can see. We're biased to finding changes in things like glutamine metabolism or serine metabolism, because these are the pathways that we're looking at when we trace glucose or glutamine. And so, but the results were quite striking, that the sources of amino acids were very different for these cells, that the epidermal stem cells really, really, really needed to have serine around if they were going to self-renew. And the post-implantation epiblast-like cells, these metastable cells, really, really, really need glutamine for their viability, but these naive cells didn't. And that began to suggest to us that maybe changes in amino acid metabolism is a really unifying feature of cells undergoing cell fate changes, and that maybe there are many different amino acids that are differentially required across these cell types. But we lacked a way to sort of systematically assess what amino acids might be important or what might not. And I know that the Plath Lab is doing really cool work in this area by adding amino acids. We actually had a very different approach where we decided to assess amino acid dependencies by removing them. So we went back to our embryonic stem cells, which we could culture in metastable conditions or naive conditions. Most of the time, these experiments are actually done with 2I on top of serum to minimize the number of variables, but all of our effects will hold whether or not serum is present. And again, just to remind you, we like this system because the cells grow at the same rate. We really obsess about this with amino acids because, of course, amino acids will be quite important for protein translation, which will gate proliferation rates. So we can culture cells in conditions that allow them to have differences in self-renewal, but equivalent proliferation rates. And then we can just systematically start to withdraw different amino acids and see what amino acids are needed. So today I'll tell you about some of the work we've done with essential amino acids. We've removed all of the essential amino acids, but in this particular experiment, we removed methionine, lysine, phenylalanine, tyrosine, or even multiple amino acids. And we found that metastable cells don't like it when you remove essential amino acids, which is perhaps not so surprising. They're essential for a reason. We cannot synthesize their carbon backbone. So if you take them away, the cells not only cannot grow, but they actually start to die and are really quite sensitive to withdrawal of many of these essential amino acids. This was the surprise to us. The naive ground state embryonic stem cells could tolerate withdrawal of these amino acids quite well. For almost every essential amino acid, not only could they tolerate the withdrawal, they could actually gain biomass. They could undergo population doublings, they could net gain biomass despite the absence of exogenous essential amino acids. The one exception being threonine, which is very cool to us because really beautiful work from Steve McKnight and Luke Cantley has shown that mouse embryonic stem cells have this enzyme threonine dehydratase that makes them exquisitely sensitive to exogenous threonine. So we see that too. This is the exception to the rule. This is the amino acid that these cells can't live without. So I hope some of you are wondering what I'm talking about at this point, because these cells, these are essential amino acids. We cannot make them, but yet we cannot gain biomass without them. So there must be a source of essential amino acids. And over the past 10 years or so, work from Daphne Barsagi, Craig Thompson, Josh Rabinowitz, many others, has shown that there is a way that we can scavenge essential amino acids from the environment in the absence of sort of free, soluble, pre-digested amino acids. And that is a process known as macropinocytosis, which is the bulk endocytic uptake of fluids, including exogenous proteins that are present at high levels in our serum, in all of our interstitial fluids, and in our cell culture media. So the way this works is the cell can basically engulf extracellular fluid, and that macropinosome will fuse with a lysosome whose high acidity leads to hydrolysis of these proteins, which can release amino acids that can activate cell growth programs. There are many cool ways to measure macropinocytosis, but one way that we particularly like is the ability to incubate cells with forms of BSA. BSA, of course, is the protein that's present in our serum and cell culture medium. This is BSA that's heavily labeled with a fluorophore, so heavily labeled that it self-quenches. But upon delivery to the lysosome, that degradation and acidity allows now the fluorophore to become fluorescent, and you can by imaging or flow or any of your favorite assays, assess macropinocytosis on a per cell basis by looking at the release of the fluorescence. So when we do this, we see that naive stem cells have massive levels of macropinocytosis. They have much higher levels of fluorescence induced by macropinocytosis, and that's completely inhibited by this pretty non-specific inhibitor IPA. We think that this, again, is cell state dependent, because if we take these pre-implantation epiblast cells and now we force them to become more post-implantation-like, sorry, wrong, it's my first time presenting this. So save that thought, sorry. First question that came to us was how to put this number in context. Is this really high or is this really low? What do these numbers even mean? And so we turned to the robust literature on macropinocytosis and pancreatic cancer, and we measured macropinocytosis in our ESCs side by side with pancreatic cancer lines that have shown to be highly macropinocytotic. And especially this PANC1 line is the example of a pancreatic cancer cell that has extremely high levels of macropinocytosis. And this ASPC1 cell is a low macropinocytosis line. What we see is actually that normal ESCs are sort of in range of these cancer cells that are the gold standard for being macropinocytotic, but the naive cells are much higher. So we do think that this is a really high level of macropinocytosis for these tiny little ES cells. And we think it's cell state dependent. So again, we can, for example, use our NANOG GFP reporter lines, and we can assess DQBSA uptake and hydrolysis in the NANOG low or NANOG high cells. We see again that the NANOG high cells are much more able to engage in macropinocytosis. And if we induce these cells to adopt a more post-implantation-like identity, if we make them into epiblast-like cells, now we really collapse that DQBSA. And so I think just to end with one or two final thoughts, I'm not convinced that this is necessarily just the pre-implantation epiblast. We have been looking at other pre-implantation lineages, and I'll show you just, for example, here, primitive endoderm lines. These are lines we got from Kat Hadjikanakis' lab, shown in orange. And all of these lines actually, again, have extremely high constitutive levels of macropinocytosis, which endows them with this very unique ability to grow really well without leucine. If you look at their growth plus leucine or minus leucine, you see that they're growing at half their normal rate, even in the absence of exogenous leucine. So I think I'm out of time. So just end with our view that metabolism changes as cells change their identity. And that those metabolic changes can be both in the modes of nutrient acquisition, how they choose to acquire nutrients. Is it transporters? Is macrophagocytosis? What nutrients they choose to acquire versus synthesize, non-essential amino acids in particular. Do you need to take up your serine, or can you make it de novo? And then what they do with them, how they break these nutrients down, like glutamine. Do you oxidize it all away, or do you stop at alpha-ketoglutarate in a way that can allow you to regulate gene expression? And we think all of these things come together. The mode of acquisition, the nutrients acquired, and what they do with the nutrients collectively results in a metabolic phenotype that's specific to that cell in that place and time, that allows them to appropriately regulate their gene expression profiles to meet whatever challenges they might need to meet. So with that, I'd like to thank everybody who helped with the work. In particular, Dave Alice and Bryce Carey, who were our initial collaborators on embryonic stem cells, along with Craig Thompson and Santosh Vardana. The epidermal work is an amazing collaboration with the lab of Elaine Fuchs at the Rockefeller University. And then all my lab members who have, the Hadjikanakis lab, who's helping us with the blastocyst work, and then members of lab, especially Pavlina Todorova, who worked on, I think, almost every project I showed you today. So thank you all very much, and I'd be happy to take any questions.
Thank you so much for such a wonderful talk, really stimulating lots of ideas. And if you have a question, you can type it into the Q&A and we will try to get to it. So I'll start with the first question. You talked about the role of alpha-ketoglutarate, and you were manipulating it potentially in different ways, one through serine synthesis pathway, another through the TCA cycle, which in theory are located in different compartments of the cell. So I was wondering if you could kind of reflect on that.
I think that's a really, really good question. So if alpha-ketoglutarate is going to do anything to gene expression, it has to get to the nucleocytosolic compartment. And you're right that if glutamine is just being oxidized in the mitochondria, that's not right at all. So the problem is easily solved in the case of serine synthesis. It's being made in the right compartment. So any alpha-ketoglutarate you're getting from serine synthesis, it's very logical to me how that could be captured by chromatin regulatory machinery, because it's either going to go there or it's going to go to the mitochondria or maybe out of the cell. The glutamine-derived alpha-ketoglutarate, it's not from serine synthesis, but rather just from changes in TCA cycle homeostasis. It's harder to understand how that gets to the nucleus. And I don't have a direct way to show this, but everything I've ever seen in embryonic stem cells, at least in our hands, tells me that that glutamine never even makes it into the mitochondria. These cells grow extremely quickly. They have to make a ton of nucleotides to fuel these rapid cell divisions. And glutamine is the obligate nitrogen donor for that. So you get your glutamine to glutamate entirely in the cytosol in embryonic stem cells. And we've seen that, that we can show genetically. That glutamate then can make alpha-ketoglutarate through transamination, again, in the cytosol. And so our best hypothesis is that alpha-ketoglutarate is probably never seeing the mitochondria. And actually, and other labs have shown that IDH can make alpha-ketoglutarate in embryonic stem cells. That would come from glucose that then exits, forming citrate in the cytoplasm, can form alpha-ketoglutarate that way. But I think overall, every time we see a shift in the alpha-ketoglutarate to succinate ratio, what that really tells me, this is a gut feeling, I don't have data for it, but my gut feeling is that tells me that your alpha-ketoglutarate isn't in the mitochondria. Because if it were, I think it would just be oxidized, or I mean, there are exceptions, right? It would likely be oxidized and you would see more of an equilibration with alpha-ketoglutarate and its downstream metabolites. But when you do see that break, when you see alpha-ketoglutarate accumulate, but everything else go down, that to me argues that there's probably subcellular compartmentalization of those alpha-ketoglutarate pools.
And when you showed the OGDH inhibition or knockdown, do you think some of that alpha-ketoglutarate is leaking out, or maybe
that's a really good question, right? So to manipulate that, we go into the mitochondria. So either it can come back out through transporters, or there might be feedback such that if you're slowing down the malate aspartate shuttle, maybe your glutamine's not even getting in the first place. Yeah.
Right. So there's a question in the Q&A about if other amino acids could buffer the deleterious effects of serine withdrawal in the cancer model.
That's a good question. So the serine synthesis makes alpha-ketoglutarate by virtue of its transamination reaction. And so theoretically, anything that would force a huge flux through transaminases in a similar direction could phenocopy that.
Great. I'm wondering, you had some really interesting inhibition of this, I think it was in the squamous cell carcinoma model, where the combination of a dietary and a knockdown of one of the TCA cycle enzymes pretty much suppressed the growth. So is there any movement toward, a cancer therapy like that?
Yeah, I think especially OGDH, there's a lot of interest in targeting that for cancer therapy. I think the genesis of that interest was probably just how much glutamine cancer cells use, and OGDH is where it ultimately gets incorporated into the TCA cycle. But in collaboration with Scott Lowe, we also showed that OGDH inhibition can reverse malignant identity in pancreatic cancer, which I think is very optimistic, that you can take this really homogeneous, mutated, terrible tumor, and if you add alpha-ketoglutarate through silencing OGDH, you can change its identity back in time. So I think other labs are interested in that context, and I believe even there's an OGDH inhibitor in phase 1 or 2 trials in pancreatic cancer. Again, maybe not for alpha-ketoglutarate purposes, but that would be interesting to see.
And then in the experiment with the pulse of glutamine withdrawal to enrich for these naive, OCT4 high cells, can you do the same thing if you inhibit enzymes in the glutamine pathway?
Yeah, that's a really good question. Probably. We haven't done that. Have we done that? We might have done something like that. But yes, I think so. I think that's what I think is so neat, especially about stem cells, is they're so heterogeneous, and it's such a great playground really to test, does my metabolic pathway actually make a difference? Because you can just look, am I shifting the balance of these cells? Am I making them more likely to self-remove, more likely to differentiate? And anything we do that would sort of make a glutamine starvation mimic experiment we would predict would have that result.
Awesome. All right, well, I think we'll close it up then. Thank you, Dr. Finley, for making time for us, for giving us such an exciting presentation, and really making us think about a lot of our own work. And we'll have to have you over to the West Coast when work travel is allowed again. So thank you.
Thank you so much. Thanks for the invitation. Right. Thanks for that.